Oh, hey. This is Michael Sinclair, one-fourth of Bobology, a research group made to look at something about genomics. Trust me, I hope this is good. So, anyways, that long part, I'm so sorry for it, but I'm trying to wrap my head around this. Trust me. So right now, I was just coding something on this amazing school computer, something called Linux, which is where I did the bulk of our research, mainly pertaining to genomics. We downloaded a bunch of different genomes from the NCBI website in order to analyze them for a greater project. What is a greater project, you might ask? I'll explain that later on. Basically, that's the main procedure for our project. However, I'm going to be discussing a few important terms because this project gets a bit head-hurting. I hate thinking about it sometimes. So I want to explain the earlier concepts like k-mers, population, and diversity in order to let you guys understand our presentation better. Stay fresh. Sorry. So, we're in my science room. Well, not my science room, my teacher's science room. I was borrowing it for now. As one part of Bobology, so today I'm just going to explain, principles of the most basic principles I need to know to understand our presentation. And I would like to show with the board and more. So continuing on, the first thing is about diversity. Why is diversity in ecosystem important? So as people, if you notice, diverse communities and people are important. Having a mixture of people, a mixture of different races, ethnicities, creates a healthier environment, creates more ideas, more circulation of ideas, and in general, more intuitive designs in our real world. In an ecosystem, something similar. You want high diversity in ecosystem. You want fungi, you want your protists, bacteria, archaebacteria, you want plant life, you want animal life, you want animals from all trees of life. And this will all contribute to a healthier ecosystem. An ecosystem with just trees isn't going to live long. So it's the helping of these different aspects of our ecosystem that are important in order to reach our larger goal. And we're going to see this through with something called k-mers. We are testing how k-mers can predict the viability, the healthiness, or the diversity of an ecosystem.
Oh, I started.
So, the first thing we're going to discuss is something called k-mers. Now, what is a k-mer, you might ask? Well, it's a DNA concept. So you know how DNA is made out of 4 different types of nitrogenous bases. Adenine and thymine, and cytosine and guanine. So, when it comes to a k-mer is basically a repeating segment of this. Now, it can be any type of combination. 4 mers, 5 mers, and, up to 6 mers. Which means that, there's 4 repeating bases or 5 repeating bases. Pretty simple. Let's say in a dog, for example, a dog DNA strand. In one of their DNA strands, they have A, T, C, T, A. Pretty basic. And in a penguin, they also have A, T, C, T, A. That means that they have the same fiber. They're matching, which means that they share a k-mer. So, one of the goals of our project was to see, can we compare the tree of life through these k-mers and look at k-mer diversity, as in, how does the amount of k-mer relations differ from each other across species? Good. Okay, so let's elaborate on the k-mer. So basically, as k-mer is, the definition is a substring of sequence. So like I said earlier with 5 mers, this is an example of something you'll see in your DNA strand. ACTG, adenine, cytosine, thymine, guanine, adenine, cytosine, thymine, cytosine. So when we're looking for 5 mers, it's 5 of these. So 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, and 1, 2, 3, 4, 5, which makes 4 of that in a given substrate. So this is an example of what a k-mer is. Now, like I said, there can be 4 mers, 5 mers, 6 mers, or gone, and so on and so forth. But we mainly use 5 mers and 4 mers. Okay. I'm going to talk about ecosystems, communities, and diversity. So this is our little chart right here. So in a given ecosystem, you have something called communities. Now, you can test the diversity in a given community. This would be referred to as local diversity, also known as alpha diversity. So this is the diversity of species living in a large community. If your community has a bunch of trees, many different animals from marsupials to amphibians to mammals to birds, different types of fungi, different types of plants, your community will have biodiversity. So, locally. However, this community might have less of that. It might just only have trees, birds, as an example. So, this is when we use something called beta diversity. Beta diversity compares different communities within a larger ecosystem versus alpha diversity. So, the beta diversity can be lower in this case because this simply just doesn't have as much as diverse as this.
That's it?
So the next thing we're going to discuss is more in depth about what beta diversity is, what affects beta diversity to become higher and lower. So first thing we have to discuss is back to our community idea. So communities will contain species, like I said. So community 1 in this example have A, B, C, D, and E, right? Community 2 has D, F, Y, D. It still contains B and D, right? So whatever something replaces in a species is called turnover, which keeps the health of an ecosystem. So when turnover is high, that means that more things are getting replaced. See here, this is an example 100% turnover. None of these species are exactly the same as community 1 originally. This has less so because there's still some that are remnants of community 1. So if you want to measure in this case, out of 5, right, we still have 2. So we need to do 3. You have 3 replaced. 3, or out of 5 species, which means that we have a 60% turnover rate, right? This is what measuring turnover is, and like I said earlier, turnover means increase in beta diversity, which means a healthier ecosystem. This is an example where beta diversity can go down, collapsibility. So, like I said here, notice how communities can become more diverse and therefore healthier. If it stays the same, this makes it more collapsible. And when a community continuously is collapsible, beta diversity goes down by a lot. So we are at Hendrickson Park right now, which is my neighborhood's local park. It's known for its large amount of animals. There's a lot of animals that live here. You have a lot of trees. Let me show you. Trees. Many squirrels that lives here. There's a hawk, a friendly neighborhood hawk, like 2 of them, that especially like to circulate around my school. And there's some squirrels here. There's a lot of bird types. So I've seen ducks, gooses, mallards, cranes, swans, pigeons, and blue jays, local sparrows, as an example. So there's also a lot of algae in our river. If you see the green, it's in a river. No, this is a bad angle. But yeah. So as we're concluding our research, this is what you want to think about, the natural habitat overall. How does it piece together? How do these communities piece together into a healthy environment? And that's basically it. So I hope you like our presentation, and I have a fantastic day, and go out to your local park to have fun. Okay, now I'm crossing the bridge. Okay, bye guys.